Welcome to the Duke Center on Addiction and Behavior Change, CABC. I'm Ed Levin, director of the center, working with folks at the Duke Institute on Brain Sciences, for Brain Sciences. This is our fall 2021 seminar series. This is the first seminar of the series. And before I introduce today's speaker, I just wanted to give you a kind of a heads up in terms of what's coming on. So next week, we have Xiangchen Li from Yale University is going to speak to us about cocaine and craving and cue and stress reward responses. And then we're going to have a series of 4 talks, Duke Nicotine Research Seminar Series. This is kind of a follow-on to the 25-year-long symposium series that Jed Rose and I put together together with other folks, including Scott Swartzwelder and others, on the Duke Nicotine Research Conference. And so this is the series which will look at interacting brain systems. Paul Meyer from Buffalo, Britta Hahn from Maryland, Edie London from UCLA, and Paul Kinney from Sinai are going to give their talks on interacting systems and nicotine effects and tobacco addiction. And then we'll continue on with some more local folks, including Alex Marshall from North Carolina Central University, Justin Sharp, and Connor O'Neill from Duke. Now, Emily Rissman is going to have her talk not on December 1st, but next semester. So that actually opens up a time if anyone has a burning desire to speak or wants to nominate someone for that date. And I am putting together the seminar series for next semester for the same offer for people who would like to speak or nominate others to speak. And also we're going to be having a February 22nd symposium on cannabis, its states of legalization, and preserving community and brain health in the face of that. So now it's my privilege to introduce our premier, our first speaker for the seminar series this fall, Dr. Jennifer Rinker, PhD. He's assistant professor in departments of neuroscience and psychiatry and behavioral sciences in Charleston Alcohol Research Center, Addiction Sciences Division of the Medical University of South Carolina. She has earned her bachelor's at Western Michigan and master's and PhD at American University of Sun Valley. And, she's going to speak to us today about divergent roles of the medial prefrontal cortex and medial dorsal thalamic nucleus in alcohol-related behaviors. Jen?
Okay. So I just want to thank Dr. Levin and Katie Healy for nominating me for this seminar series. And today I'm going to talk to you, like Dr. Levin said, about the divergent roles of engagement of the medial PFC and the medial dorsal thalamus in alcohol-related behaviors. So probably don't have to belabor this point to this crowd, but alcohol is a chronic, relapsing neurological and psychological condition. It is characterized by the inability to control alcohol intake despite often severe negative consequences. It affects around 15 million adults and remains one of the top 3 leading causes of preventable death in the United States. Binge drinking accounts for about 77% of the economic burden, and this pattern of drinking has increased by about 7% over the last 10 years. And this is mostly attributable to an increase in women binge drinking. So identifying and understanding the dysregulation of circuitry recruited during the transition from moderate to binge to dependent like drinking has been the center of much of my research since graduate school. So I think we've probably all seen some version of this schematic from Dr. Koob and Dr. Volkov. But it really hammers home the complexity of the different circuitry involved in various aspects of the binge intoxication phases, the withdrawal negative affect, and the pre-occupation anticipation craving phase of the addiction cycle. And so what this diagram really nicely demonstrates is that despite the more prominent role of a particular brain region in one phase as opposed to another, there's a high degree of interconnectedness among all of these brain areas. And so they really work in concert to sort of control the development of addiction. So we can break down this diagram into each component and look at the sort of main structures that are involved. And much of my work when I first came to MUSC focused on the medial prefrontal cortex. So that's going to be kind of where we start today. So the medial PFC, individuals with alcohol use disorder have show a range of cognitive deficits, which is indicative of PFC dysfunction. They show increased activity in the dorsal medial portion of the medial PFC in response to alcohol and alcohol-related cues. And these cues are predictive of later relapse. Additionally, alcoholics have been shown to have hyperactive ventromedial PFC during periods of abstinence. And so when we're thinking about the human medial PFC, we're thinking about the dorsal medial region here, area 32, and the ventromedial region, area 25, which corresponds in the rodent, so in the rat or the mouse, to the prelimbic and the infralimbic subdivisions in the medial PFC. Most of my talk is going to be focusing on this sort of more dorsal portion of the medial PFC, the prelimbic. So in rodents, the medial PFC has been shown to be hyperactive during ethanol withdrawal and periods of abstinence. So in 2012, Olivier George and colleagues published a paper where they showed that withdrawal, so 24 hours of abstinence from either continuous access alcohol or the intermittent access model, increased CFOS in the medial PFC. And so for purposes of this paper, they looked at both the prelimbic and the infralimbic, but they saw increases in CFOS during abstinence as opposed to while alcohol was on board. And this correlated in the number of CFOS positive neurons in the medial PFC with the amount of alcohol that was consumed. Additionally, Klonowski et al. Has shown that their chronic intermittent alcohol consumption increases the excitatory drive in the medial PFC. So they have shown that chronic alcohol drinking increases spontaneous excitatory postsynaptic currents in the medial PFC. It has no effect on inhibitory postsynaptic currents, but this change in the excitatory PFCs really shifts the drive onto the output neurons in the medial PFC towards the excitatory end of the spectrum. And then some people from our group here at MUSC, so Sven Kroner, Patrick Mulholland, and Judson Chandler, a few years ago showed that chronic intermittent ethanol vapor, so this is a model we use to induce dependence, shows signs of long-term changes in plasticity. So here you can see that there's a increase in the NMDA to AMPA ratio in CIE exposed animals, either immediately after CIE exposure or into withdrawal. And this is also demonstrated by an increase in long-term potentiation in the CIE dependent animals. And then Kristen Plyle has also shown, so just more evidence showing that, you know, different models, so chronic intermittent 2-bottle choice and chronic intermittent alcohol exposure via the vapor model. She also demonstrated that in CIE exposed animals, there was a decrease in the frequency of spontaneous postsynaptic inhibitory currents, and an increase in the amplitude of the excitatory postsynaptic currents, again, shifting the synaptic drive or the excitatory drive onto those pyramidal output neurons from the medial PFC. And then more recently, Rachel Smith and Dr. Becker from our alcohol center have shown that the prelimbic medial PFC shows CIE-induced increases in CFOS at both 10 hours into withdrawal, and hopefully you can kind of appreciate that increase from these representative figures from Rachel's paper. And this CIE induced increase or withdrawal induced increase in CFOS was sustained through 26 hours of withdrawal, which is conveniently a time point that Kristen Plyle also recorded evoked firing in the medial PFC, so in the prelimbic cortex, and she showed that there was an increase in excitability as evidenced by an increase in evoked firing at these higher current injections. So all of this kind of led us to ask, you know, whether or not these medial PFC neurons were engaged in active voluntary consumption, because, you know, all of this is looking at either withdrawal induced from drinking or withdrawal from chronic intermittent ethanol exposure. And so our group, our lab kind of turned to this Fostrap technology. So if you guys aren't familiar, the Fostrap technology is really convenient. And what we can do is give tamoxifen while the animals are drinking alcohol. And tamoxifen drives the expression of TD tomato in neurons that have a high degree of FOS induction. So we can trap these neurons that are activated in response to alcohol consumption. So what we did is we showed that not only can we trap some of these neurons, so we're getting FOS induction and increased activation in response to voluntary alcohol consumption, but we also went in and recorded from both TD tomato negative, so these are kind of our control non-activated neurons in the medial PFC, as well as the TD tomato positive, so our FOS activated alcohol responsive neurons. And what you can see is that the TD tomato positive neurons show a hyperexcitable phenotype compared to the TD tomato negative neurons in the medial PFC. So, you know, this kind of established for us that the medial PFC was really engaged during voluntary alcohol consumption, and that these alcohol activated cells show this hyperexcitable phenotype. But I think the kind of key here is that if you're looking at the medial prefrontal cortex, we know that it doesn't work in isolation, right? So we know that there is a high degree of interconnectedness between, you know, the thalamus and other regions that are involved in withdrawal and negative affect, as well as the binge intoxication, ventral striatum, of course. And so I really became interested in how these sort of interconnected regions influence the PFC or influence the different behaviors that we were looking at. And so we know obviously that the medial PFC sends dense projections to both the dorsal and the ventral striatum, as well as the amygdala and the dorsal PAG, which has recently been shown to control aspects of compulsive-like drinking behavior. But it also sends dense projections to the mediodorsal thalamus. And interestingly, the mediodorsal thalamus sends reciprocal projections back to the medial PFC. And so this is kind of this high degree of interconnectedness between these two brain areas is really what got me interested in the mediodorsal thalamus in the first place. So in addition to the high degree of interconnectedness, the mediodorsal thalamus has been shown to be involved in similar behaviors to the medial PFC, including goal-directed behaviors and cognitive flexibility. And here you can see this nice topographical representation of the projections from the MD to the different regions of the medial PFC. So this group out of France put retrograde tracing beads in the dorsomedial PFC, so the prelimbic, in the infralimbic, and then the orbital frontal cortex, which are all known to receive dense innervation from the MD. And you can see this topographical representation back here in the mediodorsal thalamus, where the central division really receives dense, sends dense outputs to the ventromedial portion. This central division sends dense outputs to the orbitofrontal cortex, and this lateral division sends outputs to the dorsomedial or prelimbic portion of medial PFC. And so, like I said, the MD is this interesting sort of higher order thalamic nuclei. It does not receive dense innervation from brain areas like sensory input brain areas. And so instead it receives input from areas like the mesolimbic reward pathway. So really is sort of poised to sort of integrate all of this information at the sort of higher order cognitive level and the sort of lower order basic reward of alcohol and drug seeking. So kind of one of the first things that we really wanted to examine was whether or not the mediodorsal thalamus looks similar to the medial PFC in terms of its CIE and alcohol drinking activation. And one of the things that's interesting is that a lot of the data that's out there on the MD and the cortex shows that they tend to fire sort of in similar patterns to each other. Originally, we thought that we would see a similar increase in CFOS at the same time point, sort of that paralleled what we saw in the medial prefrontal cortex. But interestingly, what we found when we went back, we had access to the tissue that was originally published in the Smith et al. Paper that I just showed you with the medial PFC data. But the interesting thing is that they didn't quantify any of the CFOS activation in the mediodorsal thalamus. So we went back and looked at the slides and quantified that. And so you can see we just quantified whole, we did do sub-region analysis, but for simplicity's sake, I'm just going to show you the whole MD. So we found that similar to the medial PFC, 10 hours into withdrawal, we see an increase in FOS activation, which is quantified here in this graph below. And so what you can see is that at the 10-hour withdrawal time point, the mediodorsal thalamus looks very similar to the medial PFC. But interestingly, at 24 hours into withdrawal, we see a dramatic reduction in CFOS activation where we saw an increase in the medial PFC. And so this representative figure up here is representing this 10-hour time point. And when we went in and did evoked firing recordings in the mediodorsal thalamus, what we found was that CIE had no effect at the 24-hour time point on evoked firing. So you can see this was not significantly different. There may be a slight trend, but we have a pretty decent number of animals and cells recorded in both conditions. So we don't think that this trend is going to pull out as significant even if we added more animals. Kind of take-home message is that at least in terms of CIE and CIE withdrawal induced increases in CFOS and changes in activity, they don't parallel what we saw in the prelimbic cortex and the medial PFC. You know, this got us really interested in sort of how these brain areas were functioning differently. So we went back to our Fostrap mice, and here's a sort of more descriptive diagram of how the Fostrap mice work. And we allowed them to drink alcohol in an intermittent access model, which is the same as the data that I showed you previously. And then we trapped our cells and then went in and looked at the mediodorsal thalamus. So here's the medial PFC just for comparison. And when we look at the MD, we see a high degree of FOS trapping or activation in the mediodorsal thalamus, which is really sort of most densely concentrated in the medial and central aspects. We haven't yet done the recordings in these trapped cells in the mediodorsal thalamus. That's ongoing work. So I don't have any data to show you whether or not these activated cells show differences in firing like our medial PFC activated cells do. But that's coming down the line. So the question that we had after we collected this data was really, are the medial PFC and the mediodorsal thalamus dynamically engaged during voluntary ethanol consumption? So, you know, this Fostrap data sort of implicates that active alcohol consumption does, activate at least a subset of cells in the medial PFC and the mediodorsal thalamus. But we wanted to know how these highly interconnected areas, what the pattern of activity looked like between the two, because, you know, the FOS activity sort of lends that perhaps their engagement would look similar, despite the differences in CIE withdrawal FOS activation. So in order to answer this question, we utilized fiber photometry to examine changes in calcium dynamics in freely moving mice. So I expressed Gcamp 6F, so the fast variant, in the medial PFC, so the prelimbic medial PFC, and then I implanted a fiber optic cannula just dorsal to the site of infection. And so Gcamp, for those of you that aren't familiar, is a calcium sensitive fluorescent molecule. And so when calcium binds to this permutated GFP molecule, it fluoresces in the presence of 470 nanometers wavelength of light. So in order to collect the data from these activated cells during drinking, we used this fiber photometry system, and this is just kind of a little cartoon mock-up. So we illuminated the medial PFC with both a 470 nanometer wavelength of light and a 405 nanometer wavelength of light. And this is the isosbestic control for GFP, or as closely approximated as we can kind of get. And so what this does is this controls for movement activity due to movement of the cannula within the brain. So this is really a calcium independent signal, and the 470 is our calcium dependent signal. So should reveal activity directly related to whatever the behavior the animals are performing. And then the animals were in their home cage. So we have a home cage lichometer system set up where the animals can drink from an alcohol or a water bottle. And we can time lock the licking behavior to the calcium signal and sort of line up those the behavior with the calcium activity in order to assess what the activity of the medial PFC is doing while they're actively drinking. So for these experiments, we used a modified drinking in the dark procedure. So normal drinking in the dark uses 3 days of 2 hours access, 3 hours into the dark cycle to ethanol or whatever solution you are interested in testing. And then typically on day 4, they extend that period to 4 hours. However, for the purposes of the fiber photometry recordings, we only recorded for 2 hours. And so our fiber photometry recordings always occurred on day 4 of DID drinking. So I'm not capturing the initial exposure to alcohol. This is after they've habituated to the taste and the intoxicating effects of the solution. So the testing schedule for these mice is as follows. So week 1, we recorded when they had access to to water. And so for this, what we did is on testing day, we swapped their bottles. So we just gave them another bottle of water, recorded their licks, and recorded the calcium signal in the medial PFC. And then weeks 2 through 4, we gave them access on this DID schedule, so 4 days a week, to 20% ethanol, recording on day 4 of each of those weeks. And then during week 5, we recorded when they had access to a 1% sucrose solution, just as kind of a control solution that also is rewarding and that the animals will work to acquire. So this is a video. Let's see if I can get it to play. So up here on the top, what you'll see is the Gcamp signal in green, and then the alcohol licks here in orange, and this is the mouse drinking from the alcohol solution. And what you can see is that the calcium activity ramps up dramatically just prior to the initiation of a bout for alcohol, and drops off dramatically during the consumatory phase. And this repeats a couple times over the course of the session. And this is just a couple minute clip that we took during one of the recording sessions. And you can see the fiber optic tether that he is tethered to. And so we do this in their home cage so that they're super habituated to the tethering and that they're comfortable. Mice are really sensitive to stress, so moving them just prior to doing these recordings dramatically changes their behavior. So we did this to try to get the most representative drinking behavior out of these mice. So here are just some representative traces across a 2-hour session for water here in blue at the top. So the calcium signal is in blue, and this is our normalized calcium signal. So we normalize it to that isosbestic channel that we also record from to remove any movement artifacts. And so we have our normalized calcium transients here in blue for water, and the licks for water here in orange. Calcium signal for ethanol here in green, and then the calcium signal for sucrose here in red at the bottom. And so what we can do to analyze the signal, there's a number of different ways, but for the purposes of this talk, I'm just going to focus on analysis surrounding bouts of drinking. So we can isolate the signal just before the initiation of a bout for drinking water, alcohol, or sucrose, and then just after, and sort of analyze what the signal is doing across each bout for each animal. So when we average this signal from the medial PFC, we get an average calcium trace for water here in blue. So again, this is about 30 seconds before bout initiation for water, and then about the 30 seconds following bout termination. And the bouts are sort of cut in half here because each bout lasts a different amount of time. So in order to sort of line them up, we just take the signal before bout initiation and overlay it with and juxtapose it against the signal after bout termination. So here we have our water signal in blue, and we can quantify this signal by looking at the sort of peak calcium activity, or the peak fluorescence immediately before. So that's kind of the signal right here. We can look at the minimum during, and this gives us an indication of how far the signal drops down during the consumatory phase. So we take the minimum during the calcium, or during the bout for each of the solutions. And then we can take that signal, sort of the peak signal after the termination of the bout, to get a sense of how it rebounds. So then we can overlay our trace for alcohol bouts over this. And what you can see is that in the medial PFC, there's a significant increase in calcium activity that ramps up starting about 20 seconds out from bout initiation and peaks at the beginning of the bout. The signal drops off during the consumatory phase, just as I showed you in that sort of example video, and then peaks again after the termination of the bout, and that is quantified over here in these green bars on the right. And then we can overlay our sucrose signal on top of this, and as you can see, there's an even greater increase in calcium activity preceding the bout for sucrose. It has about the same peak as we see with alcohol. And then again, there's a rebound in that activity after the consumatory phase is terminated. And again, this is quantified here in the graph on the right. And so we see a significant increase in calcium activity prior to and after the termination of a bout for ethanol and sucrose compared to water. So, this was pretty interesting to us and sort of tracks some of the other data that's available with in vivo EFIS, where we see sort of this ramping of activity prior to consumption of alcohol and, kind of drops down during the consumatory phase. So we thought, perhaps this was encoding some aspect of the reinforcing value, or this was sort of an intention signal or a decision-making signal, since we see this increase in activity prior to bout initiation. So we wanted to go in and assess whether or not we could manipulate this signal at all by, doing different things to the animal. So one of the first things that we did was examine what happens to this calcium signal when we adulterate the animal's ethanol with quinine. So quinine is a very bitter tastant, and generally speaking, when you adulterate the ethanol solution with quinine, you devalue it, and so the animals reduce their consumption. And so this is just showing you here the alcohol consumption in these animals that we recorded from during the ethanol session. And then here's their calcium signal surrounding the bout for ethanol. And again, you see kind of this ramping of activity, a peak that drops off during the consumatory phase and rebounds after. And then again, if we quantify that data over here in this graph, you'll see that same sort of V-shaped function surrounding the bouts for ethanol. When we adulterate their ethanol solution with quinine, as expected, the animals reduce their consumption of the ethanol quinine solution. And we see a reduction in the calcium activity in the medial PFC. And this is, again, quantified here in the graph on the right. So we see a reduction in the peak before and the peak after bout initiation and termination. So again, this sort of suggested to us that, perhaps the medial PFC, this is encoding aspects of the reinforcing value, which facilitates decision-making, whether the animal wants to drink or not of the solution that they're presented with. And so, if this is the case, we should be able to modulate this ethanol quinine signal by making the ethanol quinine, the impact of the devaluation a little bit less. And so one way that we can do this is by making the animals dependent. So again, all of this data I've showed you so far is in non-dependent animals that are just going through the sort of drinking in the dark binge alcohol drinking model. And so if we make these animals dependent, we wanted to know what that would look like, what their PFC signal would look like. So again, this is the same data I just showed you, just kind of truncated for simplicity's sake. We're looking at the pre-CIE, pre-bout signal. So again, we see an elevated signal for the ethanol alone solution, and we see a diminished signal for the adulterated solution, which tracks their ethanol intake. And when we make the animals dependent by putting them through multiple cycles of chronic intermittent ethanol vapor, we see that they no longer devalue the ethanol quinine solution. So suggesting that alcohol dependence has, made them tolerant of the bitter taste of quinine, or that they're so motivated to consume alcohol that they're willing to endure the bitter taste despite, the sort of aversive effects of it. And so again, our hypothesis was that if this was encoding reinforcement or reinforcing value or intention to drink in some aspect, that we would see this diminished signal in our adulterated solution should look similar to the unadulterated solution. And in fact, that's exactly what we saw. So this was kind of interesting, really kind of reinforcing to us that this was perhaps encoding some reinforcing value or intention signal to drink in the medial PFC. So there was no difference whatsoever between our baseline ethanol recordings after dependence and the ethanol quinine solution after dependence. So then the next question really was, after we got through all of this, is so how does the mediodorsal thalamus respond to alcohol? Does it mirror the signal that we see in the medial PFC, as might be suggested by our Fostrap data, where we show a significant FOS induction in both the medial PFC and the mediodorsal thalamus? So we then implanted a fiber optic cannula over the mediodorsal thalamus. And this isn't sub-region specific. Unfortunately, the mouse mediodorsal thalamus is a little bit too small to get region specificity, although we're working on recording in the circuit so that we can get a little bit of region specificity or circuit specificity. But so in this study, I injected the Gcamp virus into the mediodorsal thalamus, implanted the fiber optic cannula just dorsal to the site of infection, and then put the animals through a similar testing schedule. So we recorded when they had access to water, as well as access to 20% ethanol solution. We also adulterated their ethanol solution with quinine, and then recorded after they had access to sucrose. So if we go in and we look at these signals across all of these different sort of experimental manipulations, again, here's our average calcium trace in the mediodorsal thalamus in about the 30 seconds preceding bout initiation and the 30 seconds after bout termination. And then again, it's quantified here in this graph on the right. And if we overlay our alcohol signal, interestingly, we see a similar elevation in signal just immediately before, but it's not as pronounced in terms of the sort of ramping effect that we see in the medial PFC. But in contrast to the medial PFC, we see a massive increase in fluorescent signal or Gcamp activity after bout termination in the medial dorsal thalamus, which is in, complete opposition to what we saw in the medial PFC. And then if we look at our sucrose signal, interestingly, the medial dorsal thalamus does not respond in a similar fashion to sucrose as it does to ethanol, which is again in contrast to activity in the medial PFC, where sucrose and ethanol looked fairly similar, with sucrose eliciting even a little bit higher of calcium activity preceding and after termination of a bout. So again, we really wanted to know, okay, well, so is the medial dorsal thalamus encoding some aspect of reward or reinforcement? We know it receives inputs from the ventral tegmental area. And so we again adulterated their 20% ethanol solution with quinine. And what we see is absolutely no change in the calcium signal surrounding bouts for alcohol. And so this was really interesting to us. And so, this signal is completely unperturbed by devaluation of the alcohol solution. And so, it's the medial dorsal thalamus is responding or becoming activated to alcohol regardless of the addition of quinine. So doesn't seem to be encoding reward or reinforcing value per se, but something a little bit more specific to ethanol. And this could be due to, the pharmacological properties of alcohol. It could be due to inputs coming from other brain areas, not the medial PFC. And so this is something that we're really starting to dig in deeper to and sort of tease apart what these signals are really encoding. And so just to give you guys some appreciation of the difference in the signal between the medial PFC and the medial dorsal thalamus, here's our medial PFC signal again in solid green down here. And when I overlay the signal from the medial dorsal thalamus surrounding these bouts for alcohol, you can see the huge magnitude of difference in the signal following the termination of the bout. So we have a lot of follow-up work to do in order to tease apart what the medial dorsal signal following alcohol is really encoding. And so just to give you guys a quick summary, I've shown you guys that there is differential CFOS activation in response to CIE and CIE induced withdrawal in the medial PFC and the medial dorsal thalamus. So remember we see a decrease at the 24-hour withdrawal time point in the MD, where we see an increase in the medial PFC. Additionally, I've shown you that there are divergent effects on CIE on intrinsic excitability in the medial PFC and the medial dorsal thalamus. And so, we have these sort of starting to diverge in terms of chronic intermittent exposure to ethanol, and CFOS induction looks similar when we use the Fostrap method. So we know that voluntary ethanol consumption induces CFOS, but what we showed with our fiber photometry data is that these two brain regions, despite their high interconnectedness, show really distinct patterns of activity during voluntary ethanol consumption. So the question still remains, though, what are the functional roles of the medial PFC and the MD in alcohol drinking? So we've shown that they're activated, that they show these distinct patterns of activation, but we still really don't know quite how they're functioning to control alcohol consumption or what it is that they're encoding. So some of the studies that my lab is looking at as we move forward includes utilizing these Fostrap mice to, rather than just identify neurons that have been activated by alcohol consumption, we're actually trapping Gcamp and dreads in these neurons. So this will allow us to measure activity in highly activated cells in the medial PFC and the MD, looking at sort of how this ensemble that is activated by voluntary alcohol consumption, whether or not it is just sort of an effect of alcohol consumption or whether or not it really regulates that consumatory behavior. And so we also are really interested in utilizing an intersectional dread and fiber photometry approach. So looking at dreads in the medial PFC, and then Gcamp in the medial dorsal thalamus, and asking the question, how do medial PFC inputs to the medial dorsal thalamus influence activity during alcohol consumption? So we can record our calcium activity and manipulate this circuitry with systemic CNO. And then we can also do the reverse, where we manipulate the MD to medial PFC projections and record from the medial PFC and see how these circuits are really influencing each other. And then lastly, one of the big questions is determining the contribution of other outputs to the overall calcium signal. So the data I showed you in the medial PFC and the MD is just their CAMK2 driven Gcamp viruses. So we're looking at sort of all glutamatergic neurons in the medial PFC and the medial dorsal thalamus. And so again, some of this intersectional approach will get at how the circuit influences the downstream target. But one of the things we really want to know is how the specific outputs from the medial PFC are involved in the signal that we see in the voluntary drinking paradigm that I've showed you guys. So one of the experiments that we're working on now is looking at Gcamp activity in prelimbic to Nucleus accumbens core projections. And I can show you a little bit of preliminary data that my student has analyzed for me. And so we see a similar ramping of the Gcamp activity prior to about for alcohol in this projection-specific set of neurons in the medial PFC. So interestingly, though, this signal does not change. So I showed you guys before that the medial PFC signal is altered by things like adulteration with quinine. It looks a little bit different for sucrose. And so with these animals, we actually exposed them to chronic intermittent ethanol and compared the signal in a group of air exposed animals and CIA exposed animals, and we don't see any differences. So chronic intermittent ethanol exposure is not altering this signal in this pathway. And these studies are ongoing. So we are adulterating their ethanol solution with quinine in order to determine if this pathway is influenced by that quinine adulteration. And so with that, I'll take any questions. And sorry, I went a little bit faster than I practiced. I tend to talk fast when I'm giving talks. So I just want to do some acknowledgements really quickly. The Mulholland Lab, in particular, Patrick and Reggie Kennedy, as well as all the other members of the lab, including our two technicians who have contributed a lot to all of this data, the Becker Lab, for allowing us access to the tissue to analyze the CFOS activity in CIE exposed animals in the mediodorsal thalamus, the Woodward Lab, particularly Lac Roberts, for her help with the SLICE ePhys recordings. Obviously, all of our funding, support from the Charleston Alcohol Research Center. We're also part of the INEA Stress Consortium, and they provided a lot of resources as well. And then I just want to do a quick plug for my lab, which opened its doors on July 1st, and I am actively recruiting postdocs and trainees that are interested in fiber-based and single-cell calcium imaging approaches to explore, neurocircuitry of alcohol and alcohol stress interactions. And there are T32 spots available for support. So feel free to reach out by email if you guys are interested, and I'll take any questions.
Well, thank you so much, Jen. That was very comprehensive, very interesting, cutting-edge work, and I'm glad we have some time to discuss it. I'll lead off. I just had a question about the possible effects of cues. So it's tempting to speculate that this run-up in activity in the MD nucleus and the frontal cortex prior to the bouts is kind of like increasing urge or craving to engage in consumption. Can you test that with actually having conditioned cues and see if the conditioned cues would actually induce this this sort of response that would be might be related to craving. And in particular, I was wondering if the innervation of the MD nucleus from the ventral tegmental area was related to this increase in activity, like Gina Crowley had shown years ago with cocaine, that the increased activity in the VTA can be invoked by the display of conditioned cues.
Yeah, so to answer the first part of your question, we've actually adapted a task from Chris Lapish at Indiana University, Purdue University, Indianapolis, which is a mouthful. So he uses a task called the Two Cued Access Protocol, CABC task. And what this is animals are in operant boxes and there's a distinct auditory and visual cue for access to a retractable sipper tube that gives them water, and then a distinct set of cues that gives them access to alcohol. And we have some data that hasn't been analyzed yet from the Medial Pfc, where we've looked at calcium activity in response to the cues, as well as in response to so they get access to the sipper tube for about 10 seconds, I think, so they can drink or not as much as they want. And what we see behaviorally is a really nice discrimination between the cues. So the animals approach the alcohol bottle when the alcohol cues are played, and they approach the water bottle when the water cues are played, and they actually prefer the alcohol over the water in this setting. Interestingly, we see, sort of just anecdotally, I don't have the data fully analyzed, but from some of our initial analyses, what we see is an increase in calcium activity in response to the cue. But we see a similar response when the animal is approaching and then consuming from the alcohol bottle itself. So it's interesting because we get sort of a distinct separation in cue activation and in the medial PFC and alcohol, sort of that intention signal, if you will, that ramps up right before they initiate drinking. What we don't know is so that's in some of the earlier trials. At that point, they're fairly well trained, but we haven't put them through alcohol dependence to see if that changes the, sort of responsivity to the cue or not. But it doesn't show a shift like you see in some, other accumbens related stuff where the activity or signal shifts from the reinforcer to the cue. We see sort of distinct activation to both the cue and the reinforcer.
Okay, well, thank you. Other questions?
Yeah, I have a question. First of all, though, I would like to say that I was tickled to learn that you got your PhD with Tony Riley.
Yeah.
Because I did as well. Albeit 30 years before you, and when he still looked like Jerry Garcia.
Oh yeah, we've heard stories.
Yeah. Well, I was with him back in the day. I could probably amplify on some of those stories. But I really, his work is so elegant and just very nicely done. I really appreciated it. And I have sort of an overarching question. These regions that you're looking at, prefrontal regions and the dorsomedial thalamus, along with the hippocampus, which is my home, are all involved with memory processes as well as other processes, and are all, affected in different but interesting ways by chronic ethanol exposure and in adolescence as well. So I'm just wondering if you have any sort of general thoughts about how memory mechanisms may contribute to the development of alcohol use disorder, a shift from use to craving and so forth. Like viewing that through the memory lens, given the involvement of these structures from a memorial standpoint. I think it's an interesting take, and I'm interested in your thoughts.
Yeah, so that is an interesting question. And we've been doing more recently a lot of working memory tasks and some reversal learning stuff to look at how CIE alters medial PFC signaling in those types of tasks. So pretty simple T-maze task where they're trained to always go left to get a reinforcer, and then we do a reversal learning to have them go right. So it's a pretty simple task. And we see an interesting increase in activity in some of those kind of tasks in the medial PFC, and we haven't actually gotten there to look at it in the medial-dorsal thalamus. So I don't know how I would speculate that they would be either similar or different. But it's an interesting question, and I think it might maybe perhaps my paradigm that I'm using isn't a great one to assess that, because the animals aren't drinking chronically enough. In this drinking in the dark paradigm, it's just 3 weeks max access to alcohol for just a couple hours a day. So they're certainly not dependent, although we do see some changes in neuroplasticity over those few weeks, or sort of a start towards changes in plasticity. But I think certainly looking at some of these more chronic models, or chronic access, or doing more of the dependence exposure, we might see changes where I think sort of our working hypothesis is that the MD is sort of hyper engaged in the dependent state. And so it's actually driving them, because we know it's involved in reward choice selection or choice behaviors. And so our sort of working hypothesis is that it's hyper engaged and driving the animals towards choosing, sort of perseverating on the alcohol reward over other alternative reinforcers. And that's something that we're actively testing and might get at, sort of a perseveration of the memory of alcohol, perhaps, something along those lines. But that's really speculative on my part, and something that we're definitely going to have to test in order to make some more concrete conclusions about that.
Yeah, thank you. It's a good idea, though, and it's, I think, a valuable way to think about it.
Yeah, absolutely.
Well, thank you. Other questions or comments?
Jen, this is Scott Moore. I really liked your talk. It was fascinating.
Thank you.
I had a couple little questions about some of the early electrophysiology you showed. When you showed the pulses in response, or the action potentials in response to depolarization, was that done in the presence of any glutamate receptor blockers?
So you're talking about this data here or in the PFC?
Yeah. Oh, and stay on that slide, please. Yeah,
Okay.
but that data, yes.
So no, it was not. This was all just evoked firing, no blockers in the solution.
Okay, I think the earlier ones showed a pretty significant increase in the depolarization, which makes me think it's probably an increased resistance due to inhibition of potassium channels rather than an increase in glutamatergic afferents. But did you see any change in the width of the action potentials?
Nope. No, there was, we looked at all the different sort of aspects of the physiological signal in the MD, and we didn't see any changes in peak or Hapwidth or anything. This was the closest we got to any kind of significance, was just the slight maybe trend towards an increase in evoked firing.
Okay, great, thanks. And this, one final question, this may be totally unrepresentative because with an N of 2, we usually refer to that as case after case. But in the, I guess this is the CIE, the two spikes after the depolarizing, or after the, I'm sorry, it's after the hyperpolarizing pulse.
Yeah. So these are the rebound burst firing.
Yeah, you get pretty significant depolarizing after potentials.
Yes. So and this is something, so we're thinking about these like T-type calcium channels that are pretty densely expressed in the mediodorsal, in the thalamus in general, but in the MD, as perhaps there's some kind of CIE-induced change in expression or function of those channels, but we haven't explored that yet.
All right. Well, thank you very much. I appreciate it.
Yeah.
Good talk.
Other questions, comments?
Just leveraging off of the other Scott's comment, did you look at the after hyperpolarization durations or amplitudes? Because that could influence AP number as well, of course.
Yeah, it definitely could. And I don't recall. I think we did look at them. I don't know that we broke it down into like the sort of short, medium, and long AHPs, but I think we did not see a significant difference, if I'm recalling correctly. I'm pretty sure that Lech looked at that difference.
Yeah, I would think so. But if not, it might be worth looking at, because there are other currents that drive those AHPs that could be
Sure.
mechanistically
SK.
significant if there were a difference.
Yeah, absolutely. And we've shown differences in SK channel function in the medial PFC after alcohol exposure. So that can certainly influence the AHP. KV7 channels is another interest of ours as well, that can influence all of this evoked firing and repolarization. So
great.
Okay. Anyone else? If not, I have another question.
I just want to say, hi, Jen. It took me a minute. I was going, where do I know her from? And then when Scott
you
said
were on
Tony,
my, yep.
I was on your prelim committee. So
Yep, you were.
Wow, small world. I just want to say
I know.
congratulations on how far you have come since then. And this is
Thank
beautiful
you.
work, and nice to reconnect with you. Thank you.
Yeah, absolutely.
Thanks for
Thanks. It's
joining
good
us.
to see your face on here.
You too.
Nicole is what makes Dibs work.
Most days.
So I had a question. You just mentioned briefly about the periaqueductal gray and its connections with the MD nucleus, and thinking about George Koob and Livia George's work with the dark side. Whether usually people think about reinforcement as kind of, oh, you're going after something good, but they have made an excellent case in terms of avoiding the bad things.
Right.
So might that connection then be related to pain and negative affect induced retreat to drinking?
Yeah, so Cody Siciliano at Vanderbilt has recently shown that medial PFC projections to the dorsal PAG are involved in sort of a compulsive-like phenotype, where they continue to drink alcohol despite sort of negative consequences. And he uses a quinine adulteration model after binge drinking in order to assess that. So definitely, that could certainly be involved. I don't know about MD projections to the PAG in terms of their involvement in alcohol use disorder or sort of the negative pain or negative, phenotypes associated with alcohol consumption. So I actually don't know much of anything about that pathway. So, but certainly an interesting one to explore. And I don't know that I've seen anything in alcohol literature about that. So.
Okay. Well, thanks. And anyone else? Comments? Questions?
I have a question. I also really liked your talk. I think it's really powerful to be able to do fibrophotometry in your legometer apparatus. One question that I had is in the medial dorsal thalamus, when you had that after, the activation at the termination of a bout, that seemed like really fast, right? Like too fast to be pharmacological in nature, and it seemed to coincide directly at the termination of the bout. So I was just sort of wondering, that's super fast.
Yeah.
And you would expect it to be ramping up if it was pharmacological or even sensory. So I wonder if it is actually sort of like an action signal for bout termination. Although you don't see it with other reinforcers.
Yeah. So that's the thing, is when we look at, when you sort of overlay the sucrose and the, water onto this signal, you don't see the same pattern of activity. So I think if it was strictly a motor related thing, we would see that sort of across the board.
But what
it's really specific to alcohol? Yeah.
I'm assuming you have variability in bout size and bout duration. Do you see the same magnitude of that bout termination peak? I guess I'm calling it that now. Do you see the same magnitude of that peak, irregardless of how much they drank during a bout?
Yeah, so actually, the interesting thing is that bouts for sucrose are longer and they drink more and they lick more than they do for any of the other solutions that we give them. So again, if it was some kind of motor or signal related to, the actual licking activity, I would assume that we would see it in the water or the sucrose at a minimum.
But just within alcohol, ignoring sucrose and water for now, if you just look at the size of the alcohol bout, does that track with the magnitude of that termination signal?
So that is a good question. We could go back and look at that. We haven't looked at it for the medial dorsal thalamus. When we looked at it in the medial PFC, so that signal is obviously a little bit different, and we were more interested in what was happening sort of on the front end of things. But we did not see any correlation with peak or ramping or sort of the dip or the peak afterwards in the medial PFC that correlated with bout size or bout duration or number of licks. So it seemed to be a pretty consistent thing. We also didn't see any changes, and this is true for the MD. These mice are drinking a fairly substantial amount of alcohol. They are getting, physiologically relevant blood alcohol concentrations, and we don't see a change in like the signal surrounding the bout as they become intoxicated. So the signal is persistent despite, BECs that exceed 80 mg per deciliter.
It's super cool. It's such a powerful technique that you can answer all of these questions. It's really exciting. I look forward to seeing more of your work.
Right.
Yeah, it's been a lot of fun, and it's I'd have to say the most immediately reinforcing science that I've done, because you can like see the signal in real time as they're drinking alcohol and performing behaviors. So it's really cool to watch.
I did have one other quick question. I saw in some of your slides, in the FOSS slides, you had some activation in the paraventricular thalamus.
Oh yeah.
And so like, I'm wondering about that, because that's an important motivational area. It gets orexinergic projections from the lateral hypothalamus. So what's up with that? Why focus on the MD rather than the paraventricular thalamus?
So so we sort of honed in on the MD after we got this FOSS activation data, which again, we see activity in the PVT in the CIE animals, but we also see activation in the PVT in our controls as well. So we focused on the MD because of the sort of large distinction between our alcohol group and our control group. So that's why we focused on that. But it's interesting that voluntary alcohol consumption definitely activates the PVT, and that's actually a focus of our INEA stress grant. And so we're looking at PVT projections to the accumbens and sort of a stress alcohol interaction, looking at actually CRF projections, CRF positive neurons in that brain area. So it's definitely a interest and focus of mine. I'm become increasingly interested in these sort of midline thalamic nuclei and their role in all these different behaviors. But yeah, we didn't ignore the PVT. It's just we saw this data after the fact. So
very cool. Thank you.
Well, thanks everybody for your comments, and a very interesting talk as well as discussion. And we're well past the end of the hour, but I wanted to thank you again, Jen, for such a great talk. And if people wanted to email you, I assume that you're up for continuing the conversation that way.
Yeah, absolutely. Shoot me an email if you have any further questions. And thank you guys so much for having me. This was a lot of fun.
Yeah. And I wanted to remind everybody, next week, a week from today, September 8th, Dr. Cheng-Chan Lee from Yale University is going to speak with us about chronic cocaine craving, imaging, cue stress, and reward responses. Okay, thanks. Have a great day.
Thank you. You guys too.
Bye.